basically, a lot of us, especially working in ecology and evolution fields, are thinking about really large global scale hypotheses and how we can assemble globally sampled, deeply phylogenetically sampled biodiversity data to meet some of these challenges. And so this is a figure recently published in the Tree of Life issue of AJB on missing data patterns across the seed planes. And so even in groups that people think are fairly well understood, like angiosperms, it's surprising how much data is missing and how phylogenetically and spatially biased a lot of this is. But slowly, we're bridging some of these gaps in terms of data availability, and so the possibilities for doing really large-scale analyses are increasing, and so we're able to do things that 5 years ago we wouldn't have been able to think of. And I just want to think about, or point out that collections, and really I want to focus on natural history data in the broadest sense, not just herbarium specimens, but especially herbarium specimens, have been really integral to enabling large-scale analyses of this type. And so I think it's fair to say that largely because of herbarium digitization and other sources of natural history data, it's largely become normalized in the community to be able to do large-scale, thousands of species level attributes analyses. And so what I mean by biodiversity analyses includes not just phylogenies, as we've seen in a number of other talks in this session, but trying to link that with species-level attributes of some type. And so this can be very commonly environmental niche data, it could be trait data, chromosome numbers, genome structure, something like this, that are linked to species in a phylogeny done in a phylogenetic framework. And one of the powers of having this kind of integrated data is that we can go beyond just sort of characterizing patterns of biodiversity and maybe make some progress towards teasing apart potential causative drivers behind the patterns that we see today. So as far as the possibilities and resources available for this, you're probably familiar with quite a few of these, but for phylogenetics, there's a lot out there. So a lot of us do our own phylogenomic data collection. We could do GenBank supermatrices, and we could combine these in a number of ways. And of course, through consensus taxonomic and phylogenetic products, like OpenTree, we could generate phylogenetic hypotheses at arbitrary scales. On the ecological niche side, there are tons of tools available, huge amounts of data, the largest data providers being GBIF and iDigBio. As Pam outlined, there's tons of data layer resources that we could use to look at not just environmental or bioclimatic niche, but soil and topography and things like that. Now, what I'm going to focus on mostly for the purpose of this talk is traits, because these are data that are much more hard-won, but potentially really valuable for understanding biodiversity patterns. So I'm focusing on the clade Saxifragales. This is an order of angiosperms that's really highly diverse in terms of habitat. So we have everything from desert parasites to tropical epiphytes and rainforest trees, aquatic invasives, Arctic alpine rock dwellers. And so an amazing amount of diversity here, and a really great motivating case for trying to do a deeply sampled biodiversity data set that gets us habitat data in a phylogenetic framework, and also potentially morphological trait data to try to see what's going on with generating all this habitat diversity. So we need to think about how to get trait data at scale. And so this big book here is probably giving you a hint to what I'm going to be talking about. So this audience, I'm sure we need no introduction to what a flora is, but very briefly, it is a regional level taxonomic account that's more or less comprehensive, possibly also taxonomically or phylogenetically constrained. And so compared to herbarium specimen directly sampled data, this is fundamentally a different sort of sampling level and product. And so these are expert-derived consensus data, generally at species level, and they're largely complementary to data that we can get directly from specimens, potentially covering different sets of traits than we can get from images, and potentially a little bit more possibility to do this quite rapidly. So this is a typical herbarium, or a flora description that I got from one of the treatments in Saxifragales. And so there's a lot of data that we can see right here, and I'm going to break it down into what we can turn into biodiversity analyses. So in this region that's being covered here, we have an accepted name. So we know that this lineage occurs in the area. We could build checklists and things from this. we have synonymy data. And actually, this is often higher quality than data that's online, like the plant list. And so we've actually mined these data and merged them with the plant list to do a lot of our taxonomic reconciliation. And those of you that do large-scale biodiversity analyses will know that oftentimes that name reconciliation is a critical and very difficult step. So we also have geographic core scale information, we have habitat data, and especially this huge block of trait data. And so this kind of really broadly covers, at least in terms of diagnostic traits, the whole plant body from roots to fruits and something about life history traits. So as a test case in Saxifragales, we coded about 24 traits that were selected to be widely scorable both across taxa and across treatments. And we included basically all recent flora treatments for the Saxifragales, things like Flora Japan, Flora China, Flora North America. We did a bunch of PDF scanning and OCR recognition. And so these traits include things like, are they woody or herbaceous, something about lifespan, reproductive phenology, plant height, dimensions of a bunch of plant traits, descriptive habitat and elevation that we could combine with niche data. We've mostly been doing manual extraction for now, other than extraction of taxonomic data. And so we do plan eventually to compare this with some results from text extraction traits, but now we have a really high quality manually assembled data that we could compare that with. And my experience with this kind of makes me not sure about where the boundary lay in terms of diminishing returns, whether we want to do it sort of brute force manually or through fancy informatics methods. Because in about 3 months, with 2 really awesome undergrads on this project, we got 2,400 species scored, so covering the whole order. And so it's kind of a question of the scale that you're working on. So Saxifragales has about 2,400 species. Very briefly going through the phylogeny, we think we probably have one of the best sampled large clades of angiosperms, so we're at 72% species level coverage. This includes both phylogenomic and genetic data. And we have pretty good coverage of phylogenetic tips. After some fairly detailed cleaning steps, we have 84% coverage. Now for traits, we actually have complete coverage of species, at least for some data. And our overall matrix has about 60% data completion. I'll go through some of those missing data patterns a little bit later on. In terms of matching this to the tree, we set a cutoff based on some initial analyses of how much missing data we were willing to deal with. And we set a cutoff of at least 20% of traits sampled. And we got about 80% coverage of phylogenetic tips. So we're doing pretty similarly to our occurrence data. Now this type of data is highly multivariate, as you might imagine. And so one of the first problems is how to summarize information across dozens of variables. Not just because it's a lot of analyses, but many of these are sort of fairly holistic quantities. Certainly environmental niche is one of these, where we sort of want to know what's happening in terms of overall patterns. And so for niche data, typically if we have any occurrences at all, we can populate all of our axes. And so we can do phylogenetic PCA, no need for imputation or anything like that. Trait data, things are a little bit more complicated. So this is just, I guess, the first couple dozen or so records from our trait database. And these are a set of Adromiscus species and Crassulaceae drawn from a comprehensive family level treatment. And so in the same treatment, we have a number of patterns here. So one thing is a mix of categorical and continuous data. And in comparative data, that's often very frustrating to deal with, and especially missing data. And this is actually a single treatment, and so that doesn't, different editorial standards don't account for this, but certainly when you're combining across data sources, that's a huge problem. Also, lack of diagnostic information means it's probably not reported, and most especially, and surprisingly frequently, structures are simply absent. So if you have neuristic traits and you have no petals, you're missing data. And so we're basically compelled to deal with missing data in some way. So the way I handled this is, rather than PCA, sort of more distance-based approach to handle some of these problems a little more naturally. So it's basically a multi-dimensional scaling framework based on a pairwise distance matrix. So the distance metric is a composite one. So for categorical data, we're using the Jaccard distance you might be familiar with. And for continuous data, we're using a Euclidean distance. And we're normalizing it to a scale 0 to 1. And we're actually using equal weight for categorical and continuous data, partly because they have approximately similar numbers of axes in our data. And a very simple way that naturally handles missing data is in each pairwise comparison to drop axes only for that comparison, and then for other comparisons to drop different axes. And so basically, axes that have missing data are not voting towards the distance. So I'm just going to close the talk really briefly by going through some applications that we've been able to do. So kind of one of the first things, if we want to do comparative methods, is look for some kind of phylogenetic signal in our data. And it turns out in Saxifragales, as well as a number of other empirical data sets, these kinds of patterns are widely observable. And actually, it's fairly rare to be able to compare traits and niche at this level, and we have that happening for both of them in fairly similar patterns. And so basically, one of the interpretations you could get from that is that we may be potentially capturing at least some niche delimiting traits or their correlates. And so we're capturing a little bit of the signal of niche in our trait data. And I'll show you some more analyses that might sort of help back that up. So another thing is really strong trait-niche correlation. And so maybe not so surprising, but this gives an opportunity to be able to characterize this spatially and phylogenetically. Looking at this in a little bit of a different way, rather than phylogenetic correlation, we could characterize niche and trait disparity. So in this plot, each dot is a clade, and the metric is pairwise distances among all pairs in that clade. And we see a decent correlation and a really strong p-value for a relationship between these two, and so that's really nice to see. Now, if we want to look at this phylogenetically instead, so ask where they're mapping on a tree, these are ancestral reconstructions of the first axis of coordination for both these species attributes. And the black dots are basically the most outstanding node to parent node shifts in the ancestral reconstruction. This is actually the upper 95th percentile. And if you look at major niche shifts and you compare them to major phenotype shifts, they are highly co-localized in the phylogeny. So 60% of major niche shifts also have in the exact same place major phenotypic shifts. And if you were to kind of allow it to include neighboring taxa, then it would probably be almost all of them. We can also do things like characterize rate parameters. So we could ask about how diversification relates through time or through the tree, relate to evolution of niche traits, and we could compare that to paleoclimate data to try to tease apart biodiversity drivers. So in summary, with existing tools, we now have the ability to build really deeply sampled large biodiversity data sets and test lots of hypotheses about evolution and ecology, largely using diverse sorts of natural history data. So with that, I'd like to acknowledge my affiliation at the Soltis Lab and Florida Museum of Natural History. Numerous herbaria enabled multiple aspects of this work, both the traits and the phylogeny and the niche data. Undergraduates were instrumental, especially on the flora digitization side. So a series of those, and then NSF has kindly supported our Saxifragales for 15 years. Thanks.